Hello everyone, welcome to MMBS111, Introductory Microbiology. This is mini lecture 13.1. In chapter 13 we are discussing specific immunity, and section 13.1 is an introduction to specific immunity and that third line of defense that we mentioned in chapter 12. There are 5 learning outcomes for this mini lecture. The first is to describe how the third line of defense differs from the first and second. 2, compare the terms antigen, immunogen, and epitope. 3, list the 4 stages of a specific immune response. 4, discuss the role of cell markers in the immune response. And finally, describe the major histocompatibility complex. So, we mentioned in chapter 12 different lines of defense that are involved in the human immune system and in initiating an immune response. Chapter 13 focuses on the third and final line of defense, and this is specific immunity. So while the first 2 lines of defense are innate immunity, so you're born with these structures and forms of defense. In specific immunity, this kind of immunity is adaptive, and it's only acquired after an individual has experienced an immunizing event, like an infection or receiving a childhood vaccination. The third line of defense, or specific immunity, provides immunocompetence, and that means that the body is able to react to innumerable foreign substances that might invade the body. Because the system is adaptive, so it responds to new foreign invaders. There are 2 features that characterize specific immunity, and that is specificity and memory. So the response is specific to an antigen, and it stimulates cells that remember. So if that antigen is experienced again, cells are present that remember that antigen, and an attack is immediate. Okay. When we're talking about specific immunity, we're talking about antigens, or these are also known as immunogens, and these are substances that stimulate a response by B cells and T cells. The epitope is a small portion of the antigen that actually flags that compound or substance or object as foreign. And these can be proteins or polysaccharides that are on or inside cells, found on viruses. It can be any exposed or released protein or polysaccharide. And unlike the PAMPs that we talked about in chapter 12, antigens are very individual for each structure, and they're going to stimulate a very specific response from specific cells. There are 4 principal stages of the development of these immune cells and their interactions. And the first step in specific immunity is lymphocyte development and differentiation. So while B cells and T cells originate from the same type of lymphocyte, they are going to differentiate from one another through development in different tissues and maturation in those tissues with different chemicals. The second step in this development of specific immunity is the presentation of antigens. So antigens are going to be presented to these cells. That also is going to stimulate increased maturity and specificity for those antigens. The third step is challenging B cells and T cells with those antigens. And then finally, we're going to have differentiated cells, T lymphocytes and B lymphocytes, and they are going to respond differently because of how they have matured and developed. They respond to antigens in different ways. So the T lymphocyte response is also known as cell-mediated immunity. And the B lymphocyte response results in the production of antibodies. And we're going to look at each of these cells and their maturation and development in subsequent sections in Chapter 13. This diagram is from your text. It can be found on page 351. And this just shows you the different stages of development and maturation of B cells and T cells. So you can see that both B cells and T cells arise from the same cell type. So they originate from lymphocytes that are found in the bone marrow. B cells are going to mature in specialized sites within the bone marrow. T cells actually mature in the thymus. And then these cells are going to constantly recirculate through the circulatory and lymphatic systems. So you can see that both cell types can be found in lymph nodes and in the spleen. So they're going to migrate to those lymphatic sites. And they're going to be involved in different reactions to antigens. So B cells are primarily responsible for the production of antibodies that you're going to recognize and bind to antigens. T cells are primarily responsible for the cell-mediated immunity. They are going to stimulate other cells to respond in certain ways to antigens. Okay. So the major histocompatibility complex, or MHC, is a set of genes, and these genes code for different types of cell markers and receptors. They are found on all cells except for red blood cells, and that's because red blood cells do not have a nucleus. And these markers play a really significant role in our immune system being able to recognize and distinguish self cells from foreign substances. There are 3 different classes of genes in the MHC. The first class of genes, these class 1 genes code for the markers that are found on all of your cells except for the red blood cells, and these markers signal to your immune cells that this is self. These are self-recognition markers. The class 2 genes are going to code for regulatory markers that are found on macrophages and dendritic cells and B cells. And these class 2 genes are responsible for presenting substances that have been identified as foreign to T cells. So we're presenting an antigen on these regulatory markers that are displayed on the outside of the cell, so in the cell membrane. And the T cells are going to recognize or be stimulated by antigens that are presented in this way. The class 3 genes encode for proteins that are involved in the complement system that we talked about in chapter 12. Okay, so B cells and T cells need to be challenged or presented with antigen in order for them to differentiate into their different types. So when B cells and T cells are challenged with antigens, they proliferate, so they multiply, and they differentiate into different types and form clones. So a clone is a group of cells that are genetically identical. So we're going to end up with multiple clones of the same B or T cell that recognize the same antigen. Some of these clones will become memory cells that will ensure that when that antigen is seen in the body again, that the reaction will be rapid. The responses of B and T cells are going to differ, and as you'll see in subsequent sections here in chapter 13, we'll talk about how B cells and T cells respond differently to antigens. Okay, so cell-mediated immunity. This is the T cell response. We're going to get 3 functional types of T cells when a T cell is challenged with an antigen. First, we have helper T cells, and these T cells are going to activate macrophages. They assist B cell processes, and then they help activate cytotoxic T cells. We also have regulatory T cells, and these T cells help to control the T cell response so that it doesn't get out of control and then begin to cause damage to our own tissues. Cytotoxic T cells is our final type of T cell, and these T cells are able to destroy infected host cells, particularly host cells that are infected with virus. These are the cells that attack cancer cells. They also attack other foreign cells like bacteria. T cells secrete a number of chemicals called cytokines that help to destroy pathogens, but they do not produce antibodies. B cells are going to respond to antigen in a completely different way. So when B cells are activated by antigen, they divide and produce what are called plasma cells. Plasma cells are responsible for the production and release of antibodies into tissues and into the blood. Antibodies are protein structures that attach to the antigen that they are specific to. So an antibody this attachment and recognition is not random. There is a specific antibody that corresponds to a specific antigen, and that happens when those B cells are challenged by that antigen, and then they divide and produce more cells like them. They produce clones. Once that antibody binds to the antigen, it is marked, and those antibodies present signal to other cells in the immune system that this substance is foreign and needs to be destroyed and removed from the body. All right, so that's the end of section 13.1. Here are a few checking in questions. So make sure that you go through these and ensure that you can answer them correctly. Also, remember to go back to the beginning of the presentation and review the learning outcomes and make sure that you are familiar with those things as well. Here's our critical thinking question. So during your trip to the Dominican Republic, you are bitten by an Aedes species mosquito and subsequently transmit malaria. Malaria is caused by protists of the Plasmodium species. Since protists are eukaryotic organisms with cells similar to our own, explain how your body is able to distinguish the protist from your own cells to effectively fight the disease. So think about what we've discussed in section 13.1. Construct a good response to this question, and then you will find the answer on the next slide. So you can compare your response with mine. And hopefully this video was helpful.